Right, today's symposium. Gail Davies, which I'll have to give the talk because I say Gail's not able to be with us. We'll talk about a genome-wide association study of cognition and cognitive aging. Lars Penka is going to talk about brain imaging and cognition and speed. Alan Gao is going to talk about activity, engagement and cognitive aging. And Wendy Johnson is going to look at age 70 to age 73 changes. And here's Gail's talk. And again, forgive me, as I say, I'll just give the slides as Gail has prepared them. And here's a picture of Gail for those of you who want to see her. That's her here. And that's her with three other of the genetics team in the Lothian Birth Court, and some old bloke that came to see us last week and spent a day with us talking about genetics and cognition. I don't know if anybody recognises him, but that was him. I'll show you a younger picture when he published a single-page paper in Nature where he discovered the structure of DNA. And yeah, it's Jim Watson in the team. So, Gail's going to talk about the CAGES GWAS studies. CAGES stands for Cognitive Aging Genetics in England and Scotland, and this is a consortium. It's a consortium of 5 cohorts with longitudinal cognitive data. They're all tested on the same Illumina 610 quad version 1 with GWAS data. And again, for those of you who don't know, 610 means every individual is tested for 610,000 single nucleotide polymorphisms. They're studied for cognition in old age, for fluid and crystallized intelligence, and they're all in cohorts that study non-pathological cognitive aging. So none of these individuals is in a dementia cohort. First of all, I'm going to show you the GWAS of cognitive ability. Not cognitive aging, just the cognitive state in older age. And this is in fact just coming out. I think it's not quite, but almost available as an early PDF on molecular psychiatry, but that's the paper that Gail's presenting here first. And here are the cohorts. The so-called discovery cohorts, again, for those of you who don't know how this business works, but in genome-wide association consortia, there are usually a number of discovery cohorts, and then it moves on to a number of replication cohorts. The discovery cohorts are the Lothian Birth Cohort 1936, the Lothian Birth Cohort 1921, the Aberdeen Birth Cohort 1936, and the Downsteel DNA Bank Age and Cognitive Performance Research Centre volunteers at Manchester and Newcastle. Now, once upon a time, these were run by Pat Rabbit, so instantly you'll recognise these cohorts, some of you, and they're now run by Neil Pendleton and his colleagues in Manchester. And the replication cohort here is the Norwegian Cognitive Neurogenetics sample. Fluid and crystallised intelligence. And Gail, God bless her, has put these out for you, but everybody in the audience here will know what fluid and crystallised intelligence is. Genetics audiences don't, of course, but I'll move on. And so the analyses. First of all, for the genome-wide association study, as it says here, linear regression was used to perform association analyses using Plink. For meta-analysis of the CAGES, I'll use CAGES to describe the 5 discovery cohorts. Uses inverse standard error method used to perform the meta-analysis using metal. For gene-based analyses, and again, let me just explain that, these first analyses are done SNP by SNP. However, you can combine the single nucleotide polymorphisms into genes and do a gene-based analysis using a program called Vegas, which has a lower number of possibilities for type 1 error. In addition to that, there's a rather complicated analysis that looks at the estimation of the proportion of variance. And again, let me just read out what Gail's got here. This is a linear mixed model fitted to estimate the combined effect of all SNPs simultaneously using Peter Vischer and Jian Zhang's new method called GCTA. And what it does is it takes the genetic relationship matrix for all 3,511 subjects in the CAGES consortium and estimates and fits a linear mixed model. It looks at variance components estimated using restricted maximum likelihood, and additive variance, as it says, is partitioned to individual chromosomes. And let me just explain what's actually happening here. This is Peter's new method, which takes all SNPs in a genome-wide scan, and in these unrelated individuals, it fits a relationship matrix based on their genetic similarity and estimates using purely biological data the heritability. And then at the end there, as it says, it doesn't just estimate the heritability, it can then apportion it to individual chromosomes. And next, Gail looked at the prediction of phenotypes using only genetic information, which I'll explain later. And so first of all, here are the results. These are the genome-wide association results. And this is a Manhattan plot. And looking from, it's called that because you hope for some skyscrapers, but in many individual quantitative traits, you simply don't get the skyscrapers, and there are none here. If there were, there'd be a red dot. There aren't any red dots, so much so that I was able to put the title of the slide relatively close to the Manhattan plot because there wasn't anything standing out from the top. And so for genome-wide significance, looking for p-value of 10 to the minus 8, there is nothing for fluid or crystallized intelligence. And these are the quantile plots as well. Again, let me just point with the pointer. Up here, outside of the 95% confidence intervals, this is the expected versus the obtained p-values for all let's call it 500,000 SNPs. So there are 500,000 black dots there, and there's nothing poking far outside, as you can all see, the expected. So there's nothing genome-wide significant. There is one gene, if one does it not a SNP by SNP analysis, but a gene by gene analysis, there is in fact one genome-wide, that is a bonferroni from hell, correcting for tens of thousands of tests. And that gene is significant for fluid intelligence related to actin. Now, if you look at these quantile plots, there is something interesting about them. Now, I'm talking for Gail here and Peter Vischer, not as Ian Daly, because this is way beyond my expertise. But those black dots, quite early on, start lifting off beyond those 95% confidence intervals. Can you see that? Now, that means two things: population stratification or polygenic effects. And it's not population stratification because Peter Vischer and Gale have corrected for 4 and 20 principal components based on the GWAS to try and correct for population stratification, and it's not there. What it does mean is a polygenic effect. And so using Peter Vischer's GCTA method, for the first time, as far as we're aware, on a cognitive phenotype, we have actually been able to say how much of the genetic effect we can pick up from these purely biological SNP-based data. And I'll just turn around. And for crystallized intelligence, we've got 40% heritability. And for fluid intelligence, we've got 51% heritability. So that's a fair proportion of the genetic variance can be picked up from single nucleotide polymorphism data. What does that mean? It means that although we couldn't pick up any individual genome-wide SNP effects, it means that genetic variants in linkage disequilibrium with the tested SNPs can actually be picked up that account for a good proportion of the heritability of intelligence. In other words, this famous missing heritability is not missing. It's just there somewhere, probably in very small effects, or in copy number variants, or in rare variants, or in those sorts of things. But we have been able to show for the first time that you can pick up the heritability of intelligence, fluid and crystallized, using single nucleotide polymorphism data. And also, broadly speaking, chromosomes that are longer tend to contain more of the heritability, as you might expect. The next thing we did was this, and again, it's one of these things where you do have to hold on to your hat just a wee while to take in exactly what was done. If you just look, first of all, down to the line where it says mean, from here down to here. For the 5 discovery cohorts, we dropped one cohort out and tried to predict the intelligence of the remaining cohort, the one that's been dropped out, purely using single nucleotide polymorphism data from the other 4. You get the idea. So let's chuck one out and try and predict their intelligence just based on the SNP data of the other 4. And remember too that these cohorts have all got different cognitive phenotypes. So we've got phenotypic heterogeneity here. So we're tying our hands behind our back to some extent. What we were able to do is, with a precision of about 8 to 11%, is actually do that. So we can significantly predict intelligence on the dropped out cohort. Then our friends in Norway with the neurocognitive genetics sample said, well, we've got the same SNP data, we've got the Illumina 610 chip. Why don't you predict intelligence? So all we did to Norway was send across SNP data, and we said with these SNP data we should be able to predict the intelligence of your sample. And we did it with the same precision in the Norwegian sample as in our own cohorts, again with an entirely different set of cognitive data. Now, I do emphasize these correlations are only about 0.1, the precision is 8 to 11%. But again, Vischer, who's our statistical genetics chief, says these are remarkable data and it enables us to predict mental ability using only biological information. So, a summary of results: no genome-wide significant results observed for SNPs. QQ plots indicate deviation from the expected under the null hypothesis. Meta-analysis consistent with polygenic model of intelligence. We can account for 40 to 51% of the variation in intelligence based on common SNPs. That's actually not correct. I'm going to have to correct that line. It's by genetic variance in linkage disequilibrium with common SNPs, not the common SNPs themselves. And larger chromosomes on average explain more variation. Human intelligence is highly polygenic. A large proportion of the heritability estimate of intelligence in middle to older adulthood can be traced to biological variation using SNP data. Genetic information can be used to predict intelligence, and very large sample sizes will be needed to detect individual loci with genome-wide significance. Now, the next wee bit of the talk is newer data on cognitive aging. The discovery cohorts are the same as last time, so I'm not going to go through them. The replication cohorts are 3 Swedish cohorts overseen by Nancy Peterson, who've kindly given us access to their data and run some analyses. The phenotypes for the Lothian birth cohorts and the Aberdeen birth cohorts, it's the fluid intelligence corrected and old age corrected for age 11 Murray House test score. For Manchester and Newcastle, it's the fluid intelligence trajectory over a 15 to 20 year period. Now, we are absolutely aware that these are not the same phenotypes. That is, the change that takes place in the Lothian and in the Aberdeen birth cohorts is also present in the Manchester, but the Manchester and Newcastle is over a much shorter period of time. So once again, there's phenotypic heterogeneity that you simply can't hide away from here. The results, these again, this again is a Manhattan plot, and this time there is a dot with a red circle, which is genome-wide significant. So the previous analysis was on the cognitive state in old age. This is cognitive change. Cognitive change from either childhood to old age or midlife to old age. And we have a genome-wide significant hit. And just to show you more closely, it's significant in females but not in males at the genome-wide level. And here you can see it here. And that is, in fact, a p-value with 9 zeros after the decimal points, the kind of thing you need for these things. And what we've then done is able to take the SNPs and plot the region of chromosome 19 that's actually affected. And it's a very busy region, as it turns out. If you look down here, it's got TOM40, it's got ApoE, and it's got other genes that make a lot of sense with regard to cognitive function old age. But as bad luck would have it, this area of chromosome 19 is very, very poorly covered by our expensive 610,000 single nucleotide polymorphism chip. So what we had to do is then spend tens of thousands of more money, very importantly, carpet bombing this area and doing fine mapping of the SNPs within the area. So that's exactly what we've done. And it's really interesting, as it turns out. Oh, I should have said before that, and this is the problem. Gail has actually put in before that quite sensibly that the first thing we did was take that and ask the Swedish data to see if they could replicate it, and it was just lovely. They replicated it straight away, but not just in women, in men as well. So they've said, yes, we get that SNP, and it is significant. And again, I must emphasize, with cognitive change. Now, change is usually an unreliable variable, hard to hit, but it is significant. And then, as I said earlier, we've carpet bombed the area with fine mapping, and we found 2 different hits. This is the top SNP. We found hits on ApoE and on TOM40, and if you condition for one upon the other, Gail tells me it disappears in one direction but not in another. So we're still relatively unsure whether we've got 2 completely independent hits here for cognitive aging. So conclusions, we've got significant genetic association found with cognitive aging. First report of a GWAS of that. Replicated, which is very nice, in a meta-analysis of 3 independent Swedish populations. And the fine mapping approach finds 2 loci within the region, which we still have to do further work on. Just to finish off, 3,500 will sound like quite a large number, especially when we're replicating then in another 1,500. But in fact, it gets bigger and bigger. Gail is in fact leading, along with myself, the charged general cognitive ability. So the Lothian Birth Court 1936, just see it in the middle here, is contributing to the charged general cognitive ability consortium. So it's now a consortium that includes USA and Europe and the Antipodes, that includes over 25,000, and we're just waiting for the results from one sample to come in before we press the button on this general cognitive function genome-wide study association with reasonable power. Although the way it's getting these days, the last height genome-wide association study had 180,000 subjects in it, so 25,000 no longer seems that big. There's also a CHIC, the Childhood Intelligence Consortium, that's got 12,000 folks in it, and again the Lothian Birth Court plays a part in that, and it's got replication cohorts. And with regard to the GWAS studies that are out there, Charge decided to go first for memory, speed, and executive function, and these were all presented recently at the ICAD conference in Paris last week, and these will be coming out fairly soon. The CAGES is already coming out in molecular psychiatry, and Cogent, which I've not talked about, and Chic, which the LBC are all part of, will be coming out fairly soon. And we finally persuaded Charge to actually do general cognitive function as well, and that will be the biggest of them all with 25,000, which the LBC team is leading. And so that was Gail's talk on general cognitive function, GWAS, and cognitive aging, GWAS. Dead on time, gives you a minute to get set up, Lars. Good. And, well, just, I suppose you could thank Gail very much for her slides, and those are her acknowledgements. Thanks very much. I'm not going to take any questions.